University of Washington. So somewhat 1 year ago, we had this special number of science with the presentation of the transcriptomics, the new Atlas brain. Lein has a relevant role in this work. He's a leading researcher in the development of brain transcriptomics atlases in humans and non-human primates, and also, as we will see, in neurodegenerative disease and Alzheimer's. So please, thank you very much.
Well, thank you very much for the opportunity to speak here today, and also for the excellent talks that have already set the stage for quite a few things that I'll talk about as well. I'd really like to tell you about a slightly different perspective on brain structure and function and understanding the causes and consequences of Alzheimer's disease pathology. To begin with, let me start by saying that quite a bit of what I'm going to tell you about is based on work that's being done by major consortia funded by the US NIH, in particular the US NIH Brain Initiative and a program within the Brain Initiative called the Cell Census Project. And this has really gone through several phases. This is an attempt to bring together leading groups across the US and across the world, actually, to develop technologies for studying the brain and then apply them at scale to try to really create a complete cell atlas of the brain. The stage, which has just wrapped up a couple of years ago, was called the Brain Initiative Cell Census Network, or BTCIEN. We are now in a new phase called the Brain Initiative Cell Atlas Network, or BTCIEN, or BICAN. And this has moved from a technology development to applying to the mouse brain with piloting in larger brains, to the new phase is really now to try to go at scale to describe the human and non-human primate brain in adults and development. A couple of things I want to mention at the outset. You know, we've been really technology limited in studying the human brain. And this program has really helped to establish a new set of technologies that have opened up many new possibilities for studying the brain. Some of this is actually just moving things for brain preparation, really kind of mundane things, but can have a profound impact to improve brain banking processes and to improve the quality of tissues that can be used with modern cutting-edge histological and single-cell methods. It's also now possible to apply some of these methods to living tissues, such as neurosurgically resected tissues that are available, or in other species as well, and start to characterize the physiology and anatomy of cells with techniques like patch-seq. And then finally, understanding cells on the basis of their genetic profiles lets you also understand regulatory profiles and start to develop tools to selectively target and move from descriptive to perturbational or manipulative neuroscience as well. I won't have much time to talk about that, but I wanted to mention this descriptive work leads to new tools for being able to do experimental perturbation. The really key thing that has been touched on, but I just want to spend a little bit more time on this, is that single-cell genomics have opened up a new field here. They now allow you to measure thousands, millions of cells now from post-mortem tissues and to really handle the complexity of nervous system tissues. We're able to take tissues from cells from a model organism or nuclei from human or other species and profile many of them with transcriptomics or now with epigenomics. So really this class of technologies is single-cell genomics, and characterize all of the cell populations in these tissues. And so effectively we get a high-dimensional vector. We can cluster on that. These are putative cell types, and we can create a classification of cell types on the basis of that. And this is extremely helpful because it's a very complex tissue, but we're able to sort of bound this complexity and derive a taxonomy of cell types that now you can start to aggregate information about. You can start to understand the physiological properties of these cells, or their connectivity, or their anatomy, or their function. And so much like the genome categorize all of the genes, and we can begin to aggregate information, this is now describing all of the cells, and we can begin to aggregate their properties as well. The sort of epitome of this brain cell atlasing effort was published a few years ago based on an effort in the BTCIEN phase looking at primary motor cortex. And this really showed that this molecular technique can derive a quantitative classification of cells based on their molecular profiles, but that correlates with many other properties of cells as well. And this can be described hierarchically from a sort of very high level class to an intermediate level of granularity, the subclass, or right down to the finest granularity that one can see with these molecular methods. And by bringing different groups working on different properties of these cells, we've been able to create this census where we have the types of cells, their molecular profiles, their epigenetic profiles, their spatial organization with techniques like MERFISH that were just being described, and properties of these cells, and the richness of this annotation just keeps getting higher. Another key element of this is that this can be done in any species now. It can be done in a mouse or a monkey or a human. And in each species, you can derive a classification, and we have the tools now for doing quantitative alignment across species and identifying homologies across the species. And this is extremely important, especially for understanding the human brain. We can apply these techniques to the human brain, but we can't necessarily understand them without information that comes from model organisms. For example, we can't study developmental processes or long-range axonal projections, but we can infer them from similar studies in model organisms. And so it's both helpful to understand the human and also to understand how well other species, in fact, replicate the human or could be used as a model organism. So fast forward just 2 years beyond the single brain region, and there was a big output of the ICCN was the completion of the first atlases of the entire brain. I'm showing here this package from human and non-human primate, but in parallel, a more comprehensive atlas came out of the mouse brain describing over 5,000 types of cells in the brain. So for those of you that may be aware of similar efforts happening in other organs, this is 1 to 2 orders of magnitude higher than the cellular complexity of other organs. The brain is just simply extremely cellularly complex, and most of this is neuronal diversity. But for the purposes of this talk, it really highlighted that we can now apply these methods very routinely to human brain or to other non-human primate model organisms and achieve the similar level of granularity. The first atlas of the complete human brain has already characterized about 3,000 types of cells that we did with Sten Linneson's group from the Karolinska Institute. And that's now moved to the next stage that I won't really tell you about, but just to tell you that this is work in progress, that the ambitions for this next phase of BICAN have become much, much higher to try to tackle the scale of the problem here, to really try to get a brain-wide cell atlas of the human, macaque, and marmoset brain, to do this with both single-cell methods as well as spatial methods to get a spatial map of the tissue microorganization, to try to characterize other properties of these cells, and to link this all into a common coordinate framework that's compatible with functional imaging. So we can try to start to link functional organization with the cellular molecular organization. Okay, so all this was really a setup to tell you about the main thing I want to tell you about on Alzheimer's disease, because we've really been trying now to move beyond studying the normal brain to see if we can learn about very specific cellular consequences of Alzheimer's disease. This is sort of the intersection of 2 big consortium projects, the one that I've just mentioned in BICAN, and then the other is a new consortium formed in Seattle called the Seattle Alzheimer's Disease Brain Cell Atlas Project, where the idea is let's bring these technologies, and even a lot of crossover of the people involved in this who are creating the normal brain atlas to now start to understand what may go wrong in Alzheimer's disease. Just a little bit more detail about this. This is a collaboration between the Allen Institute and the University of Washington and Kaiser Permanente. UW and Kaiser run several Alzheimer's projects. In the case of UW, it's one of the National Alzheimer's Disease Research Centers, or ADRCs, and Kaiser runs a community study called the Adult Changes in Thought, or ACT, that can provide tissues for these studies. Now really the concept was, can we now bring together the rich neuropathology that we've been hearing about from the other talks with this new molecular information that can be gleaned from single-cell omics and spatial omics to, in a very colloquial way, understand what, where, and when things are changing in Alzheimer's disease. And just a little extra thing on this for how I sort of conceptualize this. I want to bring the resolution that we have in mouse model circuit studies to understand Alzheimer's disease. If you look in that literature, we talk about very specific kinds of cells, chandelier cells or basket cells, And we now have the tools to be able to ask what types of cells are perturbed and maybe start to understand this from the level of a circuit perspective of what are the circuit consequences that lead ultimately to cognitive decline. Okay, so let me start to describe this study in a little bit more detail. So the first element of this was really to try to improve brain collection or brain preparation methods so that we could best apply these technologies. This is work that's happening in collaboration with my good colleague Dirk Keen at the University of Washington. And for quite a number of years, we've been trying to improve how brain tissues are prepared, to improve the quality of the tissues, to improve anatomical accuracy of dissections. And we've put this to practice in this project, where he's now adopted new methods for his entire brain banking operation, but more specifically for the cohort that I'll tell you about, where this is now much better uniformly prepared slabs, improved freezing methods, much better photo documentation and things like this, which small methodological changes can make a big difference in how well this works, especially for histological quality. We've been building cohorts based on the two studies that I told you about, the ADRC, which of course is a program where typically people come in when they are symptomatic, as well as this community study where people enroll at 65 or later, not being symptomatic, and then proceed to have cognitive testing for the rest of their lives, increasingly getting additional information about those, and then donate their brains at the end. In practice, people live quite long if they have made it to 65 without dementia, so actually the average age in this cohort is 89. The cohort itself, we've picked a cohort of 84 individuals. 44 of these actually only have AD pathology, relevant for the discussions earlier, and the other half do have other comorbidities. We wanted to treat the pathology as a continuous variable. So this topic was brought up earlier about the staging. We really think that probably the local changes that we would see with omics are likely to relate to the aggregate local burden of pathology, and we need to actually capture that. But we also don't want to stage these things. We want to capture the full spread so that we can model that progression and hopefully find events related to the earliest stages. But the concept is also, let's follow the pathology. And so we are trying to follow the brain regions that have been defined previously with the typical progression of pathology across the brain. And this lets us do really two types of approaches. One is looking within a region with increasing severity of pathology, and the other is within individuals moving across the brain. I'm going to tell you about the first of these so far, where we've been looking in the middle temporal gyrus as a function of increasing disease pathology. We are trying to bring our whole suite of tools to the same cohort of individuals. So on the one hand, we have pathology, and I'll describe this in a little bit more detail. It's already been covered quite well here today, but to try to capture quantitative pathology. And on the other, to bring our toolkit of single-cell techniques. So we have single-nucleus genomics, by which I mean RNA-seq, starting to get into epigenetics, attack-seq, or the combination of the two, as well as the spatial methods now. So as was described earlier, we can not only grind this up and study the cells, but we can also study them in their native tissue context with large panels and multiplexed spatial methods. And I also want to mention that a real goal of this is a big investment to try to create these data, is to create a data resource that all of you can access as well. So on the pathology side, we are trying to use machine learning approaches to take image-based neuropathology and turn this into a quantified signal from which many features can be extracted, and then can use that to build a progression or a pseudo-progression of disease. And so this has been quite successful. We've been using the same markers that the previous talks have discussed for beta amyloid, for phosphorylated tau, alpha-synuclein, TDP43, and then really trying to quantify these more. And just on the bottom, I just want to point out here that this really is worth it because something like a Brock stage is a regional progression score with binary calls on whether or not there are any plaques or tangles in a particular region. this does not well capture the burden of pathology in a given region. And so in a given region, we see, in a given stage, we see a really wide range of pathologies for any of these markers. And so this does seem to be capturing that much better. That allows us then to create a quantitative pseudoprogression, where we're modeling this on the assumption that there would be an exponential rise in pathology over time. We use the quantitative neuropathology to do this, and I think you can see here at the top that this is really driven quite a lot by the amyloid pathology in this particular cohort. But then it correlates extremely well with other independent measurements, such as cognitive scores from CASI, ADNC scores from more stage or phasing-based measures as well. And so it does seem to be capturing in broad strokes what we had expected, but based on these quantitative measurements. So it just really, in broad strokes, this describes two main phases. An early phase where you have this linear increase in pathology. This is really preclinical at this point. And a late phase where you start to get this exponential increase in things like the number and size of plaques, for example, and much more associated with cognitive decline at this point. So we have a progression now. So then the second thing was now to map these data to find cell types from the Brain Initiative. And so for this, we've been applying the single-cell genomics techniques. We can take this Brain Initiative reference and map the Alzheimer's data to it. This then allows us to iteratively map to find cell types, the data from these Alzheimer's patients or Alzheimer's cohort. And on one hand, this lets us then map down to the finest resolution of cell types within the brain reference. In other cases, it actually has increased and expanded the reference because we have much, much better coverage of non-neuronal cells than we had in the original reference. And there are AD-specific cell states for microglia and astrocytes, for example. So at the end of the day, this creates a new reference to which all this data is mapped. It's about 139 types. In this case, this is a new term called supertype that the data can now be analyzed against. And so finally, things start to get interesting. I'll get to some results at the end here. So the first thing that we can do is simply to ask, are there vulnerable cell populations or affected cell populations, by which I mean the relative proportions of those cells have changed. So we would anticipate neuronal vulnerability as selective loss of particular types, some non-neuronal state, we might see increases in those types. And now we can look across all of these. And so I apologize for the complexity, but that's actually what we're dealing with here. It's an extremely complex system. You're seeing sort of all the various types of cells here. The inhibitory cells in this cortical region on the left, excitatory populations, non-neuronal populations, as a function of in this case, the cognitive scores or the ADNC scores, these more qualitative scores. And you can start to see that there are cell type selective populations that are affected, decreases in the neurons, increases in certain types of non-neuronal populations. But when we bring in the quantitative local aggregate pathology, now you can see those effect sizes get much larger. Furthermore, it turns out that this is a very similar pattern that's seen across cortical regions. We now looked in dorsolateral prefrontal cortex as well. And it seems to be roughly the same cell populations which are showing, vulnerability or being affected as a function of this neuropathology. So it seems to be quite robust. Furthermore, the advantage of this reference is any data set now from the community can be mapped into this in the same way as I just described our own data set. And so we've been able to map basically all other single-cell Alzheimer's studies into the same reference framework. Two of them had enough cells to actually be able to ask these same questions, and we've been able to replicate a number of these cellular vulnerability findings across studies, which I think is very promising for the reliability and robustness and reproducibility of these results. Now, so far, most of these studies have only looked at sort of control versus late stage, and you can identify vulnerable populations, but the advantage of this modeling approach is we can also identify the timing of these changes. And so here things start to get quite interesting as well. So as you might expect from the field, Some of the affected cell populations are non-neuronal. They're disease-associated microglial and astrocytic states. These are going up quite early in this pseudoprogression and continue to go up. The cells most people know about being vulnerable in Alzheimer's, the long-range projection neurons, are actually lost quite late in this progression. But surprisingly, some of the interneurons are the earliest vulnerable populations, and in particular, the somatostatin-positive interneurons in the cortex seem to show this very early loss, really when pathology levels are extremely low. And this is replicated in the frontal cortex as well. Sorry, I'm running out of time here. So we can now bring the spatial methods into play here to help to understand this better. It turns out that it's not somatostatin neurons as a whole that are affected. It's somatostatin neurons in the upper layers of the cortex that are affected. And this is in itself quite interesting because the other affected, other vulnerable neuronal populations are also in the upper layers of the cortex, but they're lost later. And so there seems to be a proximity effect that the loss of the one is then eventually leads to loss of the other populations in the same layers of the cortex. We know a lot about these cells because of the other work on characterizing phenotypic properties. So these somatostatin-positive cells are very phenotypically diverse, actually. They also include some very famous populations, such as the double bouquet cells, which are primate-specific specialization. And the vulnerable populations also have phenotypic differences from the not vulnerable populations as well. Of course, this is omics data. There's a huge amount of molecular information here for everyone to mine. These vulnerable populations also have selectively expressed genes, or that are selectively affected over the course of this pseudoprogression. I just show a few here on nerve growth factor, MME, which is neprilysin, as well, which is something which is known to be able to regulate beta amyloid cleavage. I also should mention that relin is quite selectively expressed in these somatostatin-positive neurons, in addition to the LAMP5 ones in layer 1. So I think, stepping back a little bit, we now can think of perhaps a circuit component to Alzheimer's disease, where these somatostatin neurons are the first ones lost. And what might the consequences of this be? We know a lot about this from model system research. Somatostatin neurons make inhibitory connections to all the other cells in the cortex except for themselves, so they have a very distinctive circuit property. The loss of inhibition could lead to increased excitation. Of course, it's well known that there's comorbid epilepsy with Alzheimer's disease. They're known to contribute to oscillations and communication between cortical areas. They could be trophically connected to other cells. So in fact, there is a causal relationship between their loss and the subsequent loss of the cells that they connect to. But at any rate, I think this set of techniques is starting to lead to this much higher resolution look at the disease, and potentially new targets where the cells become the targets for therapeutic intervention as opposed to a protein. I've just scratched the surface. This has characterized all of these cell populations. And so there's a really rich set of information that's now accessible from this. I can't begin to describe all of this in one thing. There will be a paper coming out shortly. In fact, it's already on bioRxiv. But the point of making this investment is to really set things up for the community to be able to mine this better and hopefully accelerate everyone's work. And so we've put a lot of effort into creating a resource for the community to work with. This has tools for being able to look at the neuropathology, at the single-cell genomics, at this trajectory analysis, and even to map your own data into this and transfer the labels onto your own data as well. So we're really trying to make this a catalytic resource that I hope will be helpful for all of you in this community. So with that, I think I'll wrap up. I'd just like to thank the many, many people involved with this. These are really big consortium projects, both within the Allen Institute itself, as well as within these other major initiatives. It takes a lot of coordinated effort to make this happen, as well as the funding support from the BRAIN Initiative, NIMH, National Institute on Aging, as well. So thank you very much.
Well, thank you very much, Ed. I'm afraid we are run out of time, so I encourage you to ask your questions to the speakers during the coffee that we have now. Thank you very much. We have had a very wonderful panel, and there is a lot to talk about now. Thank you very much. Thank you. Thank you. Sí, se nos...
The poster session now, the first one of the day, and we're going to serve some beverages and refreshments in the exhibition hall next door. I kindly ask that you return here before 12.